I'm absolutely delighted to welcome Peter Visscher and as an honorary New Amsterdammer, since he's a native son of the Netherlands. Peter is right now professor and chair of Quantitative Genetics at the University of Queensland. He's also the senior principal research fellow at the National Health Medical Research Council, and in 2010 was elected to the Australian National Academy of Sciences. So Peter is extraordinarily well respected for his work in human genetics and complex traits. His preparation for this was excellent and includes a stint looking at the heritability of this lovely herd, the Chillingham cattle, in northern England. And it turns out to have been, although you may or may not have thought it at the time, a brilliant way to segue into human genetics. Peter's path has been global. He started out in the Netherlands and then went from there to the University of Edinburgh for a PhD. This actually is hooked to the wrong, unfortunately hooked to the wrong of my animated parts. Then he traveled halfway around the world to Melbourne for his postdoc. Then he returned now as faculty in evolutionary biology at the University of Edinburgh. Then he went halfway around the world again, this time to Brisbane in 2005, and then more recently moved to the University of Queensland in 2011. And we're absolutely delighted that he's now come halfway around the world again to be able to give this talk to us. So, Peter.
Thank you very much, Pamela, for this kind introduction. And you will see my carbon print is large. I also like to thank you and Judy and Eric and Orly for initiating this wonderful new seminar series. So last week you heard from George Church giving a masterly overview of the disruptive technologies in sequencing and gene editing. And before that, Eric Green kicked off this seminar series by showing some new initiatives how to take whole genome sequencing forward, essentially, ultimately to the clinic. Both speakers gave some examples of relationships between mutations or polymorphisms and phenotypes. And in both cases, they were examples of what I call low-hanging fruit. In fact, those were the terms used. So these are where you have a mutation that is, or a polymorphism, that is sufficient to cause the phenotype. And I think this is a good paradigm for Mendelian traits, but I think it's the wrong kind of metaphor for the traits that I'm interested in, which are complex traits, by which I mean, which I include common diseases and quantitative traits. It's the wrong metaphor because this picture suggests that if our technology is good enough, for example, our ladders are tall enough, then we can just pick off the high-hanging fruit. I think that high-hanging fruit is not the right shape of what we're after in complex trait. And I think a better metaphor is one that was dreamt up by a science writer in Australia, Ella Finkel, in her book, The Genome Generation. And that's the distinction that she was trying to make there between rare variants of large effects and common variants of small effects. And she said that's a difference like having a gold nugget versus gold dust. So I think gold dust is very valuable, but it's hard to find and also hard to harvest. But I think that's the kind of right analogy here and the right metaphor for complex traits. So this is an outline of my talk. I'll start by giving you my personal overview of what I think we've learned from the genetics of complex traits in the last 5 to 10 years. Then I'll move on to something more recent, and I'd like to share with you what we've learned about looking at gene methylation from blood samples. And the thesis I'm making here is that gene methylation, so a trait that you can measure and it's a property of the genome, is just another complex trait. And then finally, I'd like to give some sort of ideas of opportunities and limitations about applying big data genomics to complex traits. So I'll start with my favorite complex trait, which is human height. The reason why that's my favorite trait is because it's easy to measure. You can get very large sample sizes. It's stable once you reach an age of 18 or so. And it's an ideal model trait because it also, as we'll see, has a high heritability. Height is also a trait that is of interest in its own right. It's associated with outcomes in life, for example, with lifetime earnings. Well, that's what my economist friends tell me. It's also a risk factor. Increased height is a risk factor for cancer. And also there's evidence just from evolutionary studies and population genetic studies that height has been under natural selection. So here we have a paper from 1886 from Francis Galton, one of these Victorian polymaths. He was the cousin of Charles Darwin. And in this, around that time, Galton and others were coming to grips with trying to quantify the resemblance between relatives for what we can now call complex traits. And here's an example from his paper where he plotted on the y-axis here the height of children against the mean of their parents on the x-axis. And he noted what he called at that time regression towards the mean, or regression towards mediocrity. That's a term he usually used. And the slope of this line is about 2/3, as Galton noted. And we now know, because the theory was developed a few decades later, that the slope of this line is one estimate of heritability. And in fact, that slope hasn't changed very much since then. Another thing you heard throughout this seminar series about precision medicine and about making predictions. Well, nothing's new there either, because Galton actually, using this information here, the regression towards the mean, and using the mean difference between males and females in height, built a prototype forecaster, a predictor of stature. So as input, we have the height of the parents, and as output is depending on whether we predict the height of a son or a daughter. Before the age of having direct measures on the DNA itself through genetic markers or sequences, we could still quantify and can quantify the proportion of variation in a population that's due to genetic factors without actually knowing how many genes are involved. And we do that by looking at the resemblance between relatives. So what I have on the top left-hand graph here is on the y-axis just a simple phenotypic correlation for different kinds of first-degree relatives. These are all first-degree relatives. And these are from 4 data sets that span more a century in terms of time. Pearson and Lee is from 1903. And these other data sets are more contemporary. And what you can see over all that time, these correlations are pretty constant, about 0.4, 0.5. What to quantify heritability, or how much variation is due to genetic factors, we usually consider different kinds of relatives. And that's shown over here. Again, I've got phenotypic correlations here on the y-axis. And on the x-axis, I have degree of relatedness. So a degree of relatedness of 1 is monozygotic twins. Degree of relatedness of a half are these guys over here, and so on. And if you do that, you can see you can easily draw a straight line through that. That is consistent with the slope of this line is consistent with most variation in a population for height being due to genetic factors. And secondly, because this line is pretty straight, it's a pretty linear relationship, that most genetic variation is additive genetic variation by nature. And you can come to the same conclusion by just doing a twin study. Here we have on the left-hand side the correlation of a bunch of monozygotic twin pairs. They're males, a very high correlation, 0.85. And on the right-hand side, we have correlation for male non-identical twins with a correlation of 0.45. And under some assumptions, we can just simply take twice the difference between these correlations and obtain a heritability of 80%. Of course, we're now in a day and age where we try to partition the total genetic variation in the contribution from individual loci. And this is to make the point that although even for a trait like height there are Mendelian or pseudo-Mendelian forms, that most variation in the population is polygenic, as shown here by this nice bell curve over here. And this is just to remind ourselves that the particular contribution from a variant, from a polymorphism or a mutation, is the combination of how frequent it is in the population and its effect size. And that's quite important when it comes also when we start thinking about genomic medicine, for example, or making predictions from DNA sequences. So points on this line, so on the x-axis here I have the frequency, and on the y-axis the effect size. And genes or variants on this curve here contribute exactly the same amount of variation in the population, or that make the same contribution to heritability. And there really are examples for many traits, in fact, where we have mutations or polymorphisms at the extreme ends of the spectrum. For example, rare variants in the fibrillin-1 gene can cause Marfan syndrome, where one of the phenotypes is that the people with this syndrome are 10 to 12 centimeters taller on average. So that's 4 or 5 inches or so. But these mutations are pretty rare. I think they're about 1 in 5,000 or so. Whereas at the other end of the spectrum, one of the first SNPs found through GWAS, genome-wide association studies, is in this gene over here. And it's an extremely common polymorphism. In fact, the frequency is more than 40%, I think, the minor allele frequency. But the effect size is tiny. It's only 3 millimeters or so per allele. But it contributes the same amount of variation in the population as this over here. So I'll jump straight to GWAS and give you my overview of the latest GWAS meta-analysis for height, which was published last year from the GIANT consortium. And this was on a discovery sample size of 250,000. So there's 250,000 people with a measure on height and a SNP chip. And what I'm showing here is how much variation is explained when you accumulate the effect of many variants. So what I have on the x-axis here is the threshold used to select SNPs in this large discovery sample. And on the y-axis I have, if we go to a new sample, how much variation, phenotypic variation, is explained by the selected SNPs. So on the left-hand side here, we have the traditional genome-wide significance level. So these are really highly replicated SNPs whose effect size is estimated without much error. And there are about 700 of those spread throughout the genome. And they explain together about 16% of phenotypic variation. So that's about, depending on what the actual heritability for height is, it's about 1/5 to 1/4 of genetic variation. If we go to the right-hand side and we say we relax our threshold of selecting SNPs in a discovery sample to 5 times 10 to the minus 3, we select about just under 10,000 SNPs, 9,500. And if we then go to a new sample and saying how much variation is explained by these selected SNPs, it's nearly 30%. This is interesting because if we don't do any SNP selection and we just ask the question how much variation in height is tagged by using all common SNPs together, that's about 45%. So this is showing that with increasing sample size, by increasing sample size, we start to get the proportion of variation explained in the selected SNPs. The signal-to-noise ratio changes so that more variation is explained by a smaller set of selected SNPs. Before I go on to prediction, and again, this is just a model, I think, for looking at height prediction as a prediction for other complex traits, including diseases. This is a nice cartoon, I think, from the Human Molecular Genetics textbook, and it's about prediction. So on the left-hand side, we have the head in the clouds, positive outlook for the future. Eureka, 21st century medicine will move from a diagnose and treat to a predict and prevent model. That's what we want, right? To the right-hand side, the head in the sand, it won't work. Most risk factors are not usefully predictive, and prediction rarely leads to an effective prevention. So with that in mind, I'll show you the result from prediction from the GIANT consortium. So again, I have on the x-axis here the threshold in selecting SNPs in the discovery sample. But now on the right-hand side, I say, what is the correlation if we go to a new sample that was not part of the discovery sample? What is the correlation between the outcome, which is the observed height, and its predicted value? And that's just a prediction from the DNA variants that a person has. And that's about 40% or so. So 0.4 is the correlation, and it's fairly insensitive to what threshold you use over here. So is that good or bad? Well, it's an awful lot better than what we had only 5 years ago, because that was probably closer to 0. But what actually does it mean? So this is okay at a population level. What does it mean for a specific individual if we make a prediction? And that's shown over here. So what I have here on the x-axis here is how good my genetic predictor is. So this is a predictor just built from genetic data. And I've parameterized that as an R-squared, so the proportion of variation in outcome, so that's in this case an observation on height, or a future observation on height, the proportion of variation in outcome that's explained by the genetic predictor. And on the y-axis I have the standard deviation of the outcome, the actual outcome, given the value of my genetic predictor. And I've parameterized that in the scale of observation, which is centimeters. So if we don't know anything, so our genetic predictor is lousy, we don't explain any phenotypic variance by our genetic predictor, the best we can do is the population mean as a predictor. And that has the population standard deviation of 7 centimeters. So that's just under 3 inches. If we look at the GWAS, the latest GWAS results from the GIANT consortium, as I told you, the proportion of variation explained there was 16%. So if we now build a predictor from that and saying how accurate is that for a specific individual, the standard deviation around the predicted value, so that would be the standard deviation of outcome given the predictor, would be still more than 6 centimeters. So we really haven't reduced that very much. It's a very inaccurate predictor for an individual. Even if we would have captured all genetic variants that influence height, so that's the heritability here, I've assumed that's 80%, and we know all the variants that have an effect on height, and we know their effect size without any error, then still our predictor would have a standard deviation of outcome around the predictor of just over 3 centimeters. And of course, that's because we can't predict the environment from a genetic predictor. So there's always residual variation there. So that's just to put the precision medicine in perspective, at least when talking about height, human height. If we think about genetic architecture, by which I mean the relationship between segregating the frequency of segregating variants in the population and their effect sizes, this is a figure we made a number of years ago, but it hasn't really changed that much. So on the x-axis here, I have allele frequency of the variant. On the y-axis, a standardized effect size in standard deviation units. So here we have rare variants with very large effects. So these are the Mendelian and pseudo-Mendelian forms of mutations that affect height. And here we have the common SNPs, very high allele frequency, very small effect size. And what is striking, I think, with this plot, and as we'll see, that's the same with when you look at specific diseases, is there's this big hole in the middle. You know, why are there no variants that have been identified for human height with a frequency of, say, 1 in 1,000 or even 1 in 100, and an effect size of, say, a few centimeters or an inch or so? And I think the main reason is we haven't, first of all, had the technology to assay all genetic variation in the population, in particular rare variation, with A, sufficient accuracy, and B, with sufficient sample size to detect these variants. But I'm pretty convinced that in the next few years, a lot of this space will be filled up. But it needs very large sample sizes. And this is my summary of partitioning the variation that we observed in human height, again, as an example for other traits. So we have 100% of phenotypic variation, and at the moment, the best estimates from pedigree data is that the heritability is about 80%. 45% of that is tagged by common variants, and of that, 16% is now explained from individual variants that are genome-wide significant. Yeah, so we're pretty sure that those specific SNPs, so one of those 700, are in close linkage disequilibrium, closely correlated with one or more causal variants that have an effect on height. So the difference between the 45% and 80% is sometimes called missing heritability, and we can speculate where that difference is. Some have argued that maybe the estimates of heritability from pedigree studies, that they're a bit overestimated, and I think probably they are a little bit. But a more interesting question, I think, is how much of this gap can we fill in by looking at rare variants? So that the explanation, that the majority of the explanation here is there are variants in the genome that are not sufficiently tagged by common SNPs. And the most likely explanation of that is that their frequency is low in the population. So we had a look at that, and that is my colleague Xiang Yang who's done this work. And we said, okay, how much variation can we actually pick up from whole genome sequence data, or in this case from imputed sequence data? So what Xiang did, he used real whole genome sequence data from the UK 10K project to simulate phenotypes. Then, after the simulation, he threw away all variants apart from SNPs that were on a bunch of commonly used SNP chips. Then he said, okay, now I've got a SNP chip, a GWAS design, let's impute those SNPs to the 1,000 genomes reference. And then finally saying, okay, how much of the original variation that I simulated from sequence data, how much can I recover? And that's what's shown on this graph over here. So on the x-axis here, I have imputation, a measure of imputation R squared with a cutoff between 0 and 0.9. And on the y-axis is an estimate of recovery, which is how much of the original variation that was simulated is recovered. And there are two groups of lines here. The ones at the top are for the common variants, which are arbitrarily defined as a frequency of 1% or more. And perhaps not surprising, most variation is captured there, nearly more than 95%. What's more interesting is when we look at the rare variants. So the rarest variant, the ones with the lowest frequency that was simulated, was of the order of 1 in 4,000 or so, a variant that occurs 1 in 4,000 copies in the data or in the population. And you can see that depending on the threshold that you put, you don't want to be too stringent in your threshold, that you recover more than 70% of that. That's quite remarkable, I think. It means that with a good imputation sequenced reference sample and a good imputation pipeline, you can recover a lot of variation. What is also very interesting, I think, with this particular simulation experiment is it doesn't seem to make an awful lot of difference which SNP chip you use, because these lines are from different SNP arrays that are all very close together. After this simulation, we were quite positive about applying this to data. So we had a data set available to us of about 50,000 individuals with SNP chip data and a measurement on height and body mass index. So we did the same, apart from the simulation, of course. We did the imputation step and then estimated how much variation do we capture from these 50,000 people for height and BMI, and that's shown over here at the bottom. And this is actually analysis where we stratify SNPs by minor allele frequency. The first thing to note is the totals that we estimate here. So the total amount of variation for height that we now capture is 60%. So we've gone from 45 to 60% by basically capturing better tagging of rare variants. The second thing is that I think that these estimates, both for height and BMI, are not that far off from the actual heritability. So in fact, there is not an awful lot that's still missing, even doing this. So this is without having individual whole genome sequence data. And the third thing to notice is that for height, it really seems that there's an excess of variation in the lowest minor allele frequency group. So that there are really rare or rare-ish variants for height segregating in the population that can be, I think, captured with sufficient sample size and genome-wide significance. So height, as I started off with, I think is a nice model trait, and now I'd like to persuade you that it is a good model, because we're now going to look at one specific disease, which is schizophrenia, and basically apply a set of the same kind of analyses. So the first one is how much variation is genetic, and looking at epidemiology and relatives. So what's shown on the top left-hand side is just simply the increased risk of first-degree relatives for schizophrenia compared to the population prevalence. And this is the graph that's in a way analogous to the phenotypic correlation for height for first-degree relatives. So a great excess here, which is consistent with genetics playing a big role. We can also look at different kinds of relatives, as we did for height. So one, again, on the right-hand side are monozygotic twins. But now, instead of a phenotypic correlation, I have the logarithm of the recurrence risk to relatives as a way to parameterize the resemblance between relatives here, the risk to relatives. And on this scale, you can see things also look pretty linear. And when you try to translate these kinds of observations into a heritability, in this case a heritability of liability, so this is a liability threshold model, for schizophrenia it also comes out at 80%, just as for height. And I'm jumping straight to the latest GWAS, which was published last year, where based on a sample size of more than 30,000 cases and 40,000 controls, about 100 individual loci were identified that are really genome-wide significant. And this is quite a landmark paper, because it wasn't that long ago, I would say 6 years ago maybe, that I would argue there was really no specific variant that people agreed with, that researchers agreed with, was robustly associated with risk of schizophrenia. And even though, as we'll see in a moment, the total amount of variation that's explained by either all common SNPs or by the genome-wide significant ones for schizophrenia is not that large, it is still sufficient at the moment already to stratify individuals by risk. So what I have on the x-axis here is individuals who are, or a scale where people are ranked from the lowest to the highest risk, just based on their SNP profile. And on the y-axis, I have the outcome, the odds ratio of risk to schizophrenia. And these are it doesn't matter which samples they are, in 3 independent samples. So what you can see here is at the 90%, so the people, the 10% highest risk, according to the SNP profile, that the actual odds ratio, when you see it, look at outcomes, is 3 to 6-fold higher. Now, this is still not huge in terms of absolute probability of getting disease, but with more data collected and more genetic variants explained, these graphs will go flat and then up much steeper, and that's for the future. And we can also look at genetic architecture. In this case, I just took that from a nice review article from Pat Sullivan and colleagues. Again, on the x-axis here, we have allele frequency, and on the y-axis, estimated effect size now as an odds ratio. And all these rare variants here are copy number variants, so low frequency in the population, but a high risk, and here are the common SNPs. And again, I would say exactly the same as I said for height. You know, where are the intermediate effect sizes with accompanied with a frequency of 1 in 1,000 or 1 in 100 or so? I think surely they must exist. And the same partitioning as we did for height, we can do for risk of schizophrenia. So 100% variation in liability. Pedigree data and twin data suggest 80% of that is due to genetic factors. We estimated a number of years ago, but there are other estimates around as well, that about a quarter of the variation in liability is tagged by common SNPs, and about just over 3% is now explained by genome-wide significant SNPs from GWAS. And just to show you that it's not schizophrenia and height, this is really a common phenomenon in whatever trait and common disease you look at. If you increase sample size, you're going to find more loci, and basically you can study more biology. So this is my simple summary from what I've learned about complex traits and common diseases from the data available only quite recently in the last 5-10 years or so. First of all, polygenicity is the norm. And by that I mean actually 2 specific things. Firstly, it's that there are variants at multiple genes that are segregating in the population that contribute to the population variation. And I think most people would agree with that. I would also argue that there is strong evidence that it's the cumulative effect of multiple variants in a person that determines their risk of disease. So that it's not that the rule is not that you need a specific mutation of a large effect to give you disease, but that it's the cumulative effect of all the risk variants that you carry that determine disease. Most genetic variation, if you just focus on the genetic variation, most genetic variation appears to be additive in nature. And I think there's actually very little variation, little evidence that individual differences, so that's another measure, of course, of variation, that individual differences between people is caused by interactions, either gene-gene or G by E. And finally, and I don't know how well accepted that is, I think it was pretty obvious, but pleiotropy is absolutely widespread, meaning that specific mutations or specific polymorphisms can have an effect on multiple traits. I don't think this should surprise us very much. This is not just what you pick up from genome-wide association studies, but if you go to other species and you look, for example, in agriculture and plant and animal breeding, it's pretty well known that there are genetic correlations between traits. And I think it's also, if you look at mouse knockouts, for example, and you study enough phenotypes, that you see that there are changes in many phenotypes for a particular mutation. And I think that was also the conclusion from the first paper from the mouse knockout project that was published in Cell a number of years ago. Okay, the second part of my talk is to share with you what we've learned from looking at DNA methylation, in particular in blood samples. So as most of you will know here, DNA methylation is one of many epigenetic regulatory mechanisms that plays a role in gene expression, in cellular differentiation, and also I think there's increasing evidence that's important in disease, either cause or consequence. And what I'm particularly interested in is also potentially a marker for environmental exposures. And this is just a little cartoon to show you that if you look at CpG islands, which are unmethylated, they don't tend to influence gene expression, whereas if it's methylated, it tends to repress gene expression. And the point I'd like to make today is that DNA methylation is just another complex trait, just as height or risk of disease. So that implies that variation between people in the specific tissue that you're studying is caused by both genetic and environmental factors that in principle you can identify. Secondly, if you use pedigree data or twin data, then you can quantify heritability for methylation. If you find that there's measures that are longitudinally stable within a person, that can be due to both environmental or what I call genetic exposures, so it can be caused by sequence variation or by some kind of environmental effect that's constant for a person over time. And finally, we can map genetic loci that influence, that's associated with DNA methylation. So the data I'll be talking about is from two different studies, and they're actually very, very different in terms of experimental design, and that makes it quite interesting. The first study is the Brisbane Systems Genomics study, which Grant Montgomery and I set up a number of years ago. It's based around families that have twins, either monozygotic or dizygotic twins, and their siblings and parents as well. We have gene methylation data on over 600 people, and gene expression data on nearly 900. And these are samples from blood. We have children here, or adolescents, that were whose samples were taken when they were about 13 years of age, and parents that were 46 years of age, or that was the median, when the samples were taken. The second set of data is from the Lothian birth cohorts, which is a study that Ian Deary at the University of Edinburgh leads. And I put Naomi here because she actually initiated the Genomics study on the Lothian birth cohort a number of years ago. So these are birth cohorts, and there are two birth cohorts, one from 1921 and one from 1936. And we had just under 1,400 individuals with genome-wide measures of methylation. And again, these are from whole blood. What's particularly interesting about this sample is that we have repeated observations. We have just under 500 people with repeated measures of methylation. For the Lothian birth cohort 1921, the baseline at which the samples were taken when these people were 79 years of age, with follow-up at 87 and 90. The younger cohort, the 1936 cohort, the mean age at baseline was 70, with follow-up at 72 and 76. And for both these studies, we used exactly the same array. It's an Illumina array to measure methylation, because that, at the time, and probably still is the best value for money in terms of getting large enough samples to make meaningful inference. So these arrays have about 450,000 CpG sites, of which there are probes there, and they're centered around genes. So Alan McRae started to look at these data and looking at the pedigree study. This is the BSGS study, and just initially quantifying on average what is the correlation between relatives. So just as we did, as I showed you for height, we can do the same for methylation. So these are average correlations for different kinds of relatives that's shown in this table here. Monozygotic twins on average, so this is across all these probes after QC. I think we had about 300,000 left or so. Average correlation is 0.2. And if we look at the average correlation of first-degree relatives, it's about half of that. And the correlation for the parent pairs is much lower, it's 0.02. So this is very suggestive of that there's genetic variation underlying these correlations. And it looks like additive genetic variation because there's a twofold difference in the correlation here. Here on the right-hand side is the histogram or distribution of the estimate of heritability from all of these probes. So just treating them like a complex trait one at a time. Most of them, as you can see, don't show any evidence for genetic variation, but there's a very long tail to the right-hand side, up to heritabilities close to 1. The second study was from the Lothian birth cohorts, where, remember, we had repeated observations. So here we can just can calculate a correlation between repeated observations, and we call that repeatability. And the histogram of or distribution of repeatability is shown here, and it looks very similar in shape to the distribution of the heritabilities in the younger people. So on the x-axis here is the estimate of correlation between repeated observations, and on the y-axis the proportion, also big spike at 0 with a very, very long tail to the right-hand side. And this circle plot on the right-hand side just shows you that the high repeatability probes are everywhere in the genome, with, as usual, although it's difficult to see, some enrichment here at the MHC locus. Okay, so we have on the one hand, in young people, we have an estimate of genetic variation of heritability, suggesting that the resemblance between relatives is partly caused by sequence variation. And on the other hand, we have in the old people, we have repeated observations, and we have a correlation between those repeated observations. So the obvious question is, are these things pointing for the same probe, are these things correlated with each other? And indeed they are. So on the x-axis here of heritability for all these 300,000 probes in bins, from 0 all the way to 1, and on the y-axis for exactly the same probes, the correlation between repeated observations in the older birth cohorts. And you can see on average there's a very, very strong correlation, which is very, very consistent with that the stability over time of those probes in the old people is just driven again by sequence variation, by heritable variation. However, there's also some very interesting examples. So here's a whole bunch of probes or CpG sites that are pretty stable within a person over a period of up to 10 years, and yet show no evidence in the pedigree data, in the family data of heritable variation. And the obvious question is, well, what could be causing those kind of correlations? So here's just one example. Here's one example of one probe where we have no evidence whatsoever of genetic variation from the pedigree data, but there's a very high repeatability correlation between repeated observations, about 0.8. And this is just shown here. Here we have the methylation measures on a scale between 0 and 1. Each point is a person, either measured here at baseline, it's wave 1, and here 6 years later or so. And as you can see, quite a strong correlation. So what could be causing this kind of correlation? Well, some environmental exposure that may be constant within a person but may vary between people. So we had some of these exposures actually measured, so we could look at that. So here's the first one where we looked at smoking as an environmental exposure. So this is now not a GWAS, although it looks like a GWAS plot. This is an MWAS, a methylation-wide association study. So it basically says at any point in the genome, looking at a correlation between a probe, between the CpG site, methylation thereof, and the trait, in this case, smoking. And what you should focus on is the scale here. When we do a GWAS with SNPs, we get quite excited if there's a point above 8 on this scale. Well, these values are are much higher. These are very, very significant associations with a not very large sample size. This is on 1,400 people. And there's lots, as you can see, there's lots of CpG sites that are highly associated with smoking. And here's just an example of one of these probes. Again, a high repeatability, correlation between repeated observations of 0.8, and also some evidence from the pedigree data of genetic variation. And this probe alone, of course, this is a highly selected one, but almost cleanly separates current smokers from ex-smokers and from never smokers. Another exposure we could look at, if you can call it exposure, is sex differences. And here are just examples of 6 different probes that are highly differentiated in a mean level of methylation between males and females. And of course, we were very keen to make sure that these probes that replicated in a different sample do not cross-bind to the sex chromosomes. So when we count up the number of significant methylation probes that we had for smoking and sex differences, we found 37 highly replicated probes that are associated with smoking, and just under 1,400, so that's very many throughout the genome, that are associated with differences between males and females. And this plot is just to show you that, in fact, these probes are not only differentiated depending on whether individuals smoked or males or females, they're also enriched for heritability and for repeatability. And I think this is mainly a reflection that there's variation there that's captured, whereas all other probes, there's a lot of them with very little variation. What I find very intriguing is when you stack up these associations and make a predictor from them, you get almost a perfect predictor. Meaning, if you were to give me a methylation profile from a new sample, I would be able to predict with very high accuracy whether that came from a person who smoked or not, and maybe even whether they ever smoked or not. And to me, that begs the question whether the association between smoking and methylation that you can pick up from the epigenetic imprint here could actually be a better predictor of subsequent risk to disease than measuring cigarette smoking itself. I don't know the answer to that, but I think it's an interesting question. The next exposure we looked at, if you can call it that way, is aging. And this follows what I think is a very nice paper from Steve Horwath from UCLA in Genome Biology 2 years ago, where he looked also at methylation array data, not just from human samples, but also from other species, and looked at their correlation between chronological age. And he came up with a concept that there may be some kind of epigenetic clock that's ticking in all of us, and that you can measure with a methylation profile, and that is pretty constant across different tissues. And here is the photograph of Steve in front of his clock. So in that paper, and also in another paper from Hannum et al. In Molecular Cell the same year, they provided estimated effect sizes of specific CpG sites and age. So we thought, okay, let's apply those predictors and see how well they do in the 2 samples that we have. And that's shown over here. On the left-hand side, on the left-hand graph, I have the actual age of a person when the sample was taken, and on the y-axis, I have their predicted biological age from the methylation profile. And this is on the left-hand side from the BSTS sample. And as I told you, there are kids in there who are 13 years of age and their parents, and you can see that the predictor really cleanly separates them. And secondly, that even within each of the 2 generations of people, there's a very good correlation there. On the right-hand side, there's actually 4 different cohorts here. 2 of them have a great spread in actual chronological age, and there's also a high correlation between the predicted age and the actual age. I'd like to now focus on these two vertical lines here, because they are from the Lothian birth cohorts, where, of course, there's very little variation in actual chronological age, because they're all born within the same year. So here we have the younger cohort, and here the older cohort in green and blue. And the intriguing question here is, well, yes, they're the same chronological age. There's a huge spread in their predicted biological age. Is this actually associated with anything? Is this does this predict anything? The fact that these individuals at the top here, they are biologically predicted much older than they really are. So something different about those people compared to those at the bottom, who are their biological age seems to be much lower than their actual age. So we associated these differences here with a subsequent mortality, because remember, we had follow-up from the baseline, because these were the predictions here were from baseline. And we found a significant and replicated association with subsequent mortality, in that, this is shown you in black here at the top, the individuals with the lowest, what we called age acceleration. So these individuals are the 25% that have the lowest biological age predicted from methylation, versus the 25% with the highest biological age. And you see there's a difference in survival there, both the older cohort and the younger cohort, and it was also found in 2 other samples. I'm not talking about causal pathways here, I'm just saying these are the observations on the associations. So the final point on methylation was that you might be able to map loci that are associated with methylation differences. Because if it's just another complex trait, we can do a GWAS. In fact, Alan McRae here ran 300,000 GWASs in the 2 samples. And this is a summary of all its results. On the left-hand side, I have for a BSGS the significant associations between SNPs and probes, and on the right-hand side for the Lothian birth cohorts. And what I have here for each of these 2 graphs, on the x-axis is the location of the probes from chromosome 1 to 22, and on the y-axis the location of the SNPs. These lines on the diagonals are not drawn lines, they are just because there's so many points there, which shows that, or implies that most of the significant associations between a SNP and a methylation differences are in cis, where the SNP is quite close to the probe. On average, they were quite close, but we found significant associations even when the SNP was about 2 megabases away from the probe. And we excluded probes here that had a sequence variant in them. And when we line up these 2 samples and look for replicated effects, we found 50,000 cis methylation QTLs, so that's quantitative trait loci, and about 2,000 trans ones that are highly significant and replicated. We can also think about prediction using methylation data. And this was inspired by a paper that came out last year in The Lancet, where this group looked at, again, an MWAS here, but now on body mass index. And they had a modest sample size. I think the discovery sample is of the order of 300 individuals or so. And they found some significant associations between methylation, again, from blood samples, and body mass index. And what caught my eye was, it's difficult to read here, but the effect sizes, to get something significant with a sample size of 350 people means that the effect sizes have to be big, and they were of the order of 1% or so. So we asked the question whether we could improve the prediction of a phenotype by combining a genetic predictor and an epigenetic predictor. So we looked at BMI and height, because that's the trait that we had available on all of these people. And we built a methylation-based predictor where we estimated effect sizes in one cohort and then applied them to a completely independent cohort, and saying how much variation in the trait is explained by the methylation predictor. And we did that in both ways. And for the SNP predictor, we used the data that I showed you from the GIANT consortium. It's actually from both for height, which was from a paper from Wood et al. Last year, and for BMI from Lockie et al., which was this year. And that's just a kind of standard profile score predictor. And the two cohorts we looked at were the Lothian birth cohorts, so that's a sample size of just under 1,400. And the other cohort is from the Netherlands called Lifelines, and these are individuals aged between 40 and 60 or so, and we had a sample size there of 450. So this is the results for BMI. So here I have the results on the left-hand side for the Lothian birth cohort, on the right-hand side for Lifelines. And there are a number of interesting things here to share with you. First of all, that the methylation score that we estimated in the other sample, so in the Lifelines, and then we applied it here, explained about 6 or 7% of variation. Yeah? So this is an association, but the estimate was from a completely different sample. And it's about the same as the genetic predictor from the GIANT consortium. Now, this green one is based on a sample size of over 300,000 people. This is based on 750 people. And the second thing that's, and it's quite similar doing it, estimating either the effect sizes in Lifelines, applying it to the Lothian birth cohort, or the other way around. And the second thing to note is when you put both predictors in, that the amount of variation you explain is basically the sum of both of them, which I think is a very interesting observation. It suggests, but doesn't prove, that what you pick up with the methylation is something to do with the environment, and not that it's the whole phenotype itself of BMI that actually creates the predictor. And it's a nice contrast to do this and then to look at height as another trait, because for height it's completely different. Basically, we don't pick up any variation that's explained by methylation differences for height. And I think that shouldn't be surprising. Height is already constant when you're, from when you're a late teenager. And why should a blood sample that's taken, 40 years later, why should that have any association with your height? And this is just showing you for completion the genetic predictor, which is very similar to what I started off with, explaining between it's about 17 or 18% of variation in these samples. Both these samples were not part of the GIANT consortium, so these are really independent. So if we then try to conclude about gene methylation and complex trait, and I hope I've convinced you that gene methylation is indeed a complex trait, and we can apply the same kind of analysis machinery to that as we did for height and for common diseases. Patterns of methylation in blood can reflect environmental exposures. I've told you about sex, age, and smoking because those are the kind of exposures that we had measured. But the question, of course, is what else can we pick up by looking at methylation? Can we, for example, pick up, say, past pesticide exposure, which is a risk factor for some neurological diseases, for example? And I think that's quite interesting to pursue. And I think, although the results I've shown you were only for body mass index, there's other papers out there in the literature which shows there is, to me, emerging evidence that methylation, even from a blood sample, which may not be the best tissue for your disease that you're studying, is actually quite useful to study either cause or consequence of disease. And even if it's just a consequence of disease, it might, I think, lead to interesting biological pathways. I think combining genetic and epigenetic information, so multi-layering of data, I think is very useful exercise. I think both in discovery, again, of discovery of genes, pathways, and networks, etc., and secondly, also for prediction. I'm not a clinician, so the potential for translation, I think, is not entirely obvious to me here, but one could just say methylation is just another biomarker, but there is some genetic variation underlying it too. Is it useful in helping with diagnosis? Or maybe with disease progression. And I think, this is early days for these kind of studies, but I think it's quite interesting data. So I put future here. This is just a couple of slides to go with a question mark, because we all know that making predictions is hard enough, and predictions about the future is even more difficult. And also, I think when you think about predicting what's going to happen in 5 to 10 years' time, if we'd gone back 10 years' time and we'd looked at all the discoveries made driven by enormous advances in technology, would we really have predicted where we are now? Well, I wouldn't, and I don't know whether many others would have. I think I'm reasonably confident in saying that whole genome, let's call them correlation studies, where we correlate polymorphisms, variants in the population with outcomes, will continue, and probably with sample sizes into the millions. I think the dichotomy between having either a SNP array or doing whole genome sequencing will disappear because of technology. I think that genetic or sequence information will not be limiting. And in fact, from the study I showed you, from the simulation study on the whole genome sequences, I think for complex trait it's hardly limiting at the moment. Of course, it's limiting if you're looking for de novo variants. Then you need to have the full information. I think there'll be so much data that the trick will be to ask the smart and the right question of the data, and to have the right analysis tools to address those specific questions. What I think will be limiting in the future is to have deep phenotyping on very many samples. So by which I mean to having multiple levels of data, not just genetic data and a phenotype or a diagnosis, but having data like gene expression, methylation, metabolomics, etc., etc. And I think that there's really power in having these data sets talk to each other. I think it's also very important to have longitudinal data. In particular, if you could have data at baseline before an individual presents with a disease. I think we need much more measures of environmental exposures, because only then we can start to impute those exposures in other samples where we haven't measured them. And of course, clinical information as well, because for these large studies we dealt with today, the phenotype is basically the most common denominator. This is just a comment. I think if we're serious about the environment and studying the environment in G/E studies, in particular modifiable risk factors, we need to measure the environment. And maybe one route of doing that is by using smart technology, smartphones and smart devices. I think for the future we'll need to combine different sources of information for both diagnosis and prediction. I personally think that sample size will keep trumping technology, by which I mean that we're not going to get away from the fact that the association between a sequence variant or a polymorphism in the population and our complex trait, that the effect sizes of that association are very, very small, and I think they'll remain small. So we just need large sample sizes. And finally, for common diseases and quantitative traits, even if we have very large discovery samples for prediction, for example, I think the outcomes will remain probabilistic, and we just have to live with that also in a clinical setting. I don't think that's going to change. So finally, I've tried to acknowledge the people who did all the hard work throughout this talk, and here are most of them again. And in particular, thanks to my collaborators in our center who did the work on the complex traits. My collaborators on the quantitative and population genetics, Mike's in Melbourne, Naomi in Brisbane, Bill Hill in Edinburgh, and Alkes in Boston. And collaborators on the methylation work from the University of Edinburgh, and from Groningen in the Netherlands, and from NHLBI, Mike Mendelson and Daniel Levy from the Framingham study. And last but not least, it's a real privilege for a complex trait geneticist like me to be able to work with some research consortia that have collected very, very large samples of data and are happy to share that for research purposes. Thank you.
All right, so thank you, Peter, for a fabulous talk. And questions? same format as usual. There are microphones on both sides. Yeah, it's on. Yep. And we've been asked to sort of move towards the middle.
Yes, hello, over here.
Oh, yes.
Hey, I'm Johan Björkegren. I work with cardiovascular disease and genetics and functional genomics. So I was very fascinated about your talk, and I've been thinking about these issues a lot. And the first question is just to understand you correctly, that most inherited variants of methylation is then explained by DNA variation, whereas you think that the variation of methylation that are not explained by DNA variation is probably environmental. So is that a correct interpretation? At least for height, then. Or?
Yeah. So well, if you look at methylation as a trait in itself, as the phenotype, then on average we found that about 20% of that is probably due to sequence variation. However, it really changes. It varies from which particular site you look at. I think because we just happen to have blood samples available, a lot of the CPG sites don't show any variation between people. So they're more or less everybody's either unmethylated or methylated there. Of those ones where we found genetic variation, well, that varies from 10 to 100%. But clearly, there's also environmental variation, and some of that seems to be at least either stochastic or random, or it's associated with the exposures that we measured.
And did you have a chance to look at how much of that sort of environmental part that could also be explained by gene expression variation?
We have started to look at that, and we find, as others have reported, that the relationship between gene expression and methylation is not straightforward. It's not always the same direction. In fact, what we found is that for those combinations of gene expression and methylation probes, because there was also array data, that are most highly phenotypically correlated with each other, that was all driven by genetic variation. Okay. But those are the ones that are easiest to pick up.
I see. And then I had a question. Height is a continuous variable, so you can measure GWAS for that. But for coronary artery disease, you have case control studies, and I've been thinking, what are the effects and interpretation of the genome-wide significant risk loci you find, given the fact that a lot of the controls will also be suffering from the same disease, but not really have developed the clinical manifestation. To my, thinking about this, my interpretation would be that you capture variants that are most important for early development of these complex diseases rather than late. Do you have any thoughts on this?
I think this will be trait dependent. So if you look at schizophrenia as an example, then the estimates of how much of the variation is genetic is pretty high, and I think what people contribute to the GWAS data are not particularly early or late onset diseases. This may be different for cardiovascular. I don't know. It really depends on the definition of the phenotype that goes into the genetic studies, I think.
Yeah, and the last final one then. So I'm looking sort of on the other side, trying to figure out what these GWAS significant loci, what's happening, what are they affecting, and we are seeing a lot of both cis and trans effects, and also I've been thinking about, do you think these variants also could have different effects, even opposing effects over time, and how would that sort of affect? I mean, given coronary artery disease, for instance, if you have a susceptibility for inflammation, you might not have increased susceptibility from the start of your life, but eventually, you get to the point when inflammation plays a role, all of a sudden you get those kind of dynamics. Have you been thinking about those, or how that could affect the measurement of the missing heritability, for instance?
I think I understand your question. So, and it's a very interesting one to think about. Genetic variation of a particular phenotype over time. For example, I've had discussions with colleagues about the heritability of body mass index. If you look at the results from twin studies, it suggests that the heritability of body mass index, so twins obviously at the same age, is very high, almost like that of height. Whereas if you look at sort of more, if you look at the GWAS results or more cross-sectional, you go across ages, the heritability seems to be much lower. Is that because body mass index when you're age 20 is not the same trait as when you're 50? It's possible that there are real interactions with age there. But to pick up interactions, the problem with interactions, to pick them up statistically, we need even larger sample sizes than we need to pick up main effects.
Okay, thank you. Very interesting talk.
Menachem?
Hi, so thanks for the great talk. In one of your figures, you showed that in the Lothian birth cohort, the higher variance in the predicted age seemed to be associated. That higher variance was associated with mortality ultimately. Why in the first cohort, I think it was the red one, I can't remember what it was called though, was there not a larger variance there also? Why did they have a better fit for the prediction, suggesting that for them mortality was less of an issue?
Okay, well, I'm not sure whether this entirely addresses your question, but because the birth cohorts, are of the individuals from across the two cohorts are very different age. We had many more people that died on the older cohorts from 1921 than from 1936. So that explains at least some of the observed variation where we fit a proportional hazards model or whatever. So that may be part of the explanation.
Thank you.
Andy?
Hi, very nice talk. I actually had two questions, one of which was about the studies where you were looking at the mortality of the individuals based on their age, based on the methylation studies. Is there any indication that the people were sick and then they died, or did they just sort of drop dead?
No, that's a very good question. Yeah. So we did an analysis where we had sort of lag time to make sure that there was no short time span between the measurement and when they died, and the results didn't really change. And we also, what I didn't say, we adjusted, for example, for cell counts that we had measured. But I also must stress that we can't, it's an association analysis, so it can't really exclude that there are other confounders that we didn't measure.
Right. Okay, so a question about the methylation studies in terms of the different kinds of probes that are on the array, right? So a lot of them are associated with CpG islands, but a lot of them are also, some of them are distributed like in gene bodies and across intergenic areas and stuff like that. And so it's, it's maybe more of a comment than a question, but it'd probably be very interesting to look at differences in those different subsets. For example, when we were looking at this a number of years ago, we definitely saw areas in gene bodies where there was methylation that was associated with genotypes that could be seen even when comparing different kinds of tissues to one another. So it looked like some kind of measure that would not be able to be determined even if you had really good RNA sequencing data. By looking at that data, you'd have to look at the methylation differences to see these signs of the genetic changes.
So we did look at the annotation that we had on the sites themselves, and to see, for example, whether any differences on average in genetic versus environmental variation, or the associations with the environmental exposures, or even the methylation QTLs. There were some differences between the different kinds of probes that are on these arrays, but they weren't huge, I don't think, at least when we looked at them.
Okay, thanks.
Judy?
Peter, great talk. So regarding that hole, for the rare variant hole between common variants and Mendelian forms, just conceptually it makes sense that it's got to be filled in. But as you get to kind of outbred populations with larger and larger sample sizes, how are you thinking about that in terms of correcting for population substructure as you kind of get to rare variants?
I think there's a couple of comments on the large sample sizes. If you look at those plots both for height and schizophrenia, and so you have some points over here, some points over there, it's easy to draw a straight line through that, of course, but that straight line would actually predict that you need the same sample sizes as we have from GWAS for, for sequencing or having these rare variants. So that's point one. The second point is, I think the biggest issue with having these huge sample sizes is that it usually comes from different sources, like a consortium, and that although people do their best, with QCing and all that, there is heterogeneity that's creeping in because, just errors are made that you can't pick up from QC. So I think what that means is that the overall, sort of, if you can think about how much genetic variation is in this huge sample of which you may only have a summary statistic, that's reduced a bit. And I think that's an issue. So it's always better, I think, like the PGC is doing, to work with individual level data because you can do so much more QC and model fitting there. With population stratification, again, in particular, as you say, for the rare variants, you'd have to cater for that. But I think that is, in principle, that's possible to do that. But I think the sample sizes in the first place and depending on if you have individual level data or summary data, the QC and the heterogeneity might be a bigger problem.
But philosophically or theoretically, you don't see any problem potentially for, if you start correcting for population substructure too finely, that somehow you're going to reach the limits of rare variant detection. You don't see a problem with that at all.
Well, I think the limits are essentially by how many copies do you see and what is the real effect of that, of that variant. But yeah, of course, when you take it to the extreme and you only see it once or twice, I mean, that's probably not good for a statistical treatment. Yeah.
Okay, thank you.
That was a wonderfully instructive talk. I'm wondering, I was particularly struck by the smokers and how you can detect based on methylation. So I'm wondering if you could tell which kind of cigarette and whether electronic cigarettes would give you a footprint.
Okay, that's a good question, but these are from old people in Scotland, and electronic cigarettes had not been invented yet, to my knowledge. But it's a very interesting question. I think that's a reason why we having to start measuring these environments or environmental exposures on large samples, and I'm hoping that maybe smart devices are the way to do it, because only once you've got it measured you can test for an association with methylation.
What about the content of the cigarette?
Excuse me?
Could you tell based on the content of the cigarette which kind they smoked? Methylation.
We've not looked at that. I don't think we have that information in the database. I don't think so, but it's an interesting one. I mean, I'm not sure whether you're thinking about it, but a potential application would be in forensics here, where you find a tissue sample and you can tell the brand of cigarette that the person smoked.
Thanks.
Eli?
Hi, I have a question about the methylation prediction of BMI and the implications for sort of methylome architecture. And that is that you were doing each cohort as discovery and then predicting the other one, right? But one cohort was like twice the size of the other one. And the R-squared that you got from the predictor was about the same in the two different cohorts. And I would have thought with the larger discovery cohort, you might expect a larger variance explained in the target set. And so, especially given that you have small sample sizes, maybe looking 5 to 10 years away, you might say that we should have tens of thousands of individuals with methylation data, but does this speak to that at all in your mind?
Yeah. No, that's a very good question, and what I forgot to say is that we had a step before we made the predictions of the estimated effect sizes, where we had a completely independent set of a few thousand individuals who were completely independent of these two cohorts. So that sample size was the same for both of them. We just used, so we selected probes from a sample out there and then took the probes and estimated the effect sizes in one and applied them to the other and the other way around. So you're right. But I do think from looking at the results from correlation with age, chronological age, and also with smoking and other traits, that there are clearly large effects there, but I wouldn't be surprised if you ramp up the sample sizes, you find a very, very long tail on small associations, suggesting that it's really affecting many sites in the genome, some of these exposures.
Hi, wonderful talk. I have a question about methylation clock and Steve Horvath's stuff. Originally, when Steve published this data, it was on a lot of tissues, and it was about 300-something sites, which predicted the biological age. And then he actually said that it might be more specific in individual tissues, and a lot of people, including me, gave him some very specific cohorts. I had a neuronal sample, and he showed that 53 CPG sites was very enough to very accurately predict age. So when we stick that to gene ontology analysis, neither these 350 CPG sites, neither these 53 sites showed any enrichment in any biologically important pathways. And we are still scratching our head. What does that mean? I'm sure you had time to think about it.
Well, I think, excuse me, I agree with that we have to be careful with causality here. It is not impossible that the methylation is just associated with something else that we haven't measured. Yeah. If not, it's telling us something new about aging, which I think is interesting. I mean, I thought the most interesting thing from Steve Horvath's work was that, he looked at his predictor. In fact, it's built from across tissues, as far as I understand it. So, which is very different from the other predictors that are around, which are usually just from blood samples, and which are also of the order of 100 or so, or even fewer sites. I don't know. The obvious thing is to study those specific sites, maybe in an experimental setting, and saying, are these themselves, directly affected by the aging process, or are they correlated with something that we haven't measured? I don't know what the answer there is, but it's a good question.
Oh, it looks like the last question is Eric's.
All right. Yeah, I think mine's sort of a related question. With the methylation set that you have and all the variables that are scored there, have you sort of tried to characterize the correlation structure among those and organize the correlation structures into modules and potentially even use the MQTLs as a perturbation source to start looking at causal inference? Yeah. And has anything useful come out of that?
Right, that's a whole bunch of questions. So I'll tell you one thing we did was to say, what is the, we have 300,000 measures after QC, of methylation at CPG sites. We know that the ones that are physically within 200 base pairs of each other or so are highly correlated, but there will be correlations elsewhere. When we estimate the effective number of independent probes, this is just from looking at the variance overall, the effective number is of the order of 200 or 300 only, rather than 300,000. So there is a very high level correlation structure there. The only thing we've looked at with QTLs is looking at when there's a high correlation between gene expression and methylation, and that seems to be driven by common, usually cis effects. That's all we've looked at so far.
All right. Well, thank you, Peter.